What's up, lads? This is your boy Jace, and welcome to the Big Brain Busters. So in this video, we're going to be talking about the glycolysis pathway. But before I begin that, allow me to just course you through, a brief overview of what glycolysis is. So glycolysis is a series of metabolic pathways that are involved in breaking down glucose into pyruvate. So glucose is a 6-carbon molecule, and it is broken down into a 3-carbon molecule, which is pyruvate. So in this case, you expect that if you are breaking down a 6-carbon molecule into a 3-carbon molecule, you expect to yield 3-carbon molecules. So in this case, you are breaking down glucose into 2 pyruvates. With that being said, allow me to also proceed by saying that glycolysis occurs in the cytosol of the cell, and the energy that is yielded from glycolysis is in form of NADH, okay, NADH and ATP. So these are the energy forms of that are produced in the glycolysis pathway. Okay, cool. So now let's jump into it. So as you can see, this is a series of 10 steps like that. So this, we're going to talk about each step one by one. But then before I begin that, this series of 10 steps is actually divided into 2 phases. So we have the investment phase and the payoff phase. So in the investment phase, from the word itself, investment, so it's like you invest into something in order to yield more. So in this case, in the glycolysis pathway, we are investing 2 energy molecules, which is 2 ATP. So we use up 2 ATP in order to produce 4 ATPs and 2 NADH. Okay, I hope that makes sense. So the investment phase is from steps 1 to 5, and then the payoff phase is from steps 6 to 10. Okay, so let's begin. So in the first step, it involves conversion of glucose into glucose 6-phosphate. And what happens here is that the enzyme hexokinase adds a phosphate on what? On the 6th carbon of glucose to produce glucose 6-phosphate. And in this case, since we are using up a phosphate, so that phosphate is actually coming from an ATP molecule. So 1 ATP molecule is used up in this step. In the next step of glycolysis, it involves conversion of glucose 6-phosphate into fructose 6-phosphate. So fructose 6-phosphate and glucose 6-phosphate, these guys are simply isomers of each other. So in this case, since they are isomers, you actually expect an isomerase enzyme to be involved in conversion of glucose 6-phosphate into fructose 6-phosphate. So the enzyme here is simply phosphohexoisomerase, and this guy, it is actually isomerized to this guy. Cool. Step 3. So in step 3, it involves simply fructose 6-phosphate being converted into fructose 1,6 bis phosphate. Sorry, it was supposed to be bis phosphate. So this guy, in this case, it involves simply addition of a phosphate group on the first carbon of fructose 6-phosphate. So in this case, after we add that phosphate, and we know that transferring of a phosphate group requires a kinase. So in this case, the enzyme that is involved is phosphofructokinase. And here again, since a phosphate has been used up, so meaning we expect that 1 ATP molecule has been used up. So that's where now in the investment phase, we have 2 ATP molecules actually used up. Then in step 4 involves conversion of fructose 1,6 bisphosphate into 2 sugar molecules. So in this case, it is converted into dihydroxyacetylphosphate and glyceraldehyde 3-phosphate. So these guys are simply, this guy is a ketone compound, and this guy is an aldehyde compound, and the enzyme that is involved in here is simply idolase enzyme. Cool. Then in the next step, which is step 5, in step 5 it involves conversion of dihydroxyacetyl phosphate into glyceraldehyde 3-phosphate. I know, right? So here we've got now, we simply have 2 G3P. I like abbreviating them like that. So we have two G3Ps. Okay, so in this case now, henceforth in this reaction, we expect that whatever will be happening this side will also be happening this side, because we've yielded two things. So I guess now this makes sense why we will form two pyruvates. Okay, so in the next step, it involves conversion of sorry, I forgot this guy. So in this So this guy are simply isomers of each other, okay? So since they are isomers of each other, so you expect an isomerase enzyme again. So the enzyme here is simply triose phosphate. Then the next step involves glyceraldehyde 3-phosphate being converted into 1,3 bisphosphoglycerate. And in this case, what is happening is that we are just simply adding a phosphate group to this compound. And since we are adding a phosphate group, in fact, we're not even actually adding a phosphate group. What we're simply doing is removing a hydrogen group. And since we are removing a hydrogen group, what will happen is that whenever you remove a hydrogen group, you need a proton carrier. So that proton carrier is simply NAD plus. So NAD plus will receive that proton, so meaning that there is yielding of what will produce NADH. And since we have a series of two steps occurring at the same time, so meaning we are producing two NADH, and the enzyme that is involved here is simply glyceraldehyde 3-phosphate dehydrogenase. A dehydrogenase removes a hydrogen. Hope it makes sense. Then in this step, it involves 1,3 bisphosphate being converted into 3-phosphoglycerate. So as you can see, we had two phosphates, and then we've removed the first phosphate on the first carbon. We've removed one phosphate and then converting it into 3-phosphoglycerate. So in this case, what the enzyme that you actually you're actually expecting to catalyze this reaction, you expect a kinase. So in short, the enzyme is simply phosphoglycerate kinase, and in this case 1 ATP is produced. Then in the next step we are actually, this one I call it the simplest and useless step but anyways, don't call it that. So in this step, we are actually involving transferring of a phosphate group. We are not even actually removing it, we're just shifting it. So it involves shifting of a phosphate group from the third carbon to the second carbon. So we are converting 3-phosphoglycerate into 2-phosphoglycerate, and in this case the enzyme that is involved is an isomerase, but we won't call it an isomerase because it's a type of an isomerase and it is known as a mutase. So the enzyme is phosphoglycerate mutase, and the phosphate is shifted as I've explained. Then the next step, it involves 2-phosphoglycerate being converted into PEP, or phosphoenolpyruvate. So the enzyme that is involved in here is simply enolase, and what happens is that we are removing a water molecule from here. So in short, since remember again, I'm continuing to emphasize on that, since there are a series of two steps occurring, so meaning that two water molecules are actually removed. Okay, then the last step involves phosphoenolpyruvate being converted into pyruvate. Okay, so in this case, what is happening is that we actually remove a phosphate from phosphoenolpyruvate, and when we remove a phosphate, simply we are producing 1 ATP. So a pyruvate kinase is actually involved in this step. So in this case now, I guess you can, from the calculations, we produced 1 ATP here, but then since we have two reactions occurring at the same time, so I mean 2 ATPs, even here 2 ATPs, so altogether in the payoff phase 4 ATPs are yielded, and then somewhere here we produced an NADH, which is a proton carrier, so meaning that we expect that we produced 2 NADHs. So the net, the gross energy molecules that have been produced are 4 ATPs and 2 NADHs. But then if we removed the 2 ATPs that we used up this side, so meaning that we are actually having 2 ATPs and 2 NADHs in the glycolysis pathway. So thank you very much for watching. Please check out our other videos on lipid metabolism and the like. Don't forget to subscribe and like and share the video. Thank you.